Hello, and welcome to the October 5th, 2017 OTCQX U.S. Company Investor Conference. My name is Bob Power with OTC Markets Group. Before I introduce the next presentation, a few quick points. Please submit questions in the small Ask a Question box on the bottom right of the screen. Also, after the presentation and question and answer session, don't log out. You'll automatically be transported to the Dyadic booth, where you can continue the conversation via the chat box and access additional investor materials. On a final note, all of today's presentations are recorded and are available for 24/7 replay at your convenience. At this point, I'm very pleased to welcome Mark Emelfarb, Chief Executive Officer and founder of Dyadic International. Welcome, Mark.
Thank you, Bob. Good afternoon, and thank you for joining today's conference call to discuss Dyadic's business plan and progress update on our C1 gene expression platform. On slide 2 is our safe harbor regarding forward-looking statements. Certain statements contained in this conference call and slide presentation are forward-looking statements within the meaning of the federal securities laws. On slide 3, I would now like to take the time to introduce you to our company, Dyadic International, Inc., our C1 gene expression platform, which is based on the fungus Mycephalothora thermophila and nicknamed C1. C1 are cell factories which we program with DNA, genes, to produce proteins in large quantities at low cost. C1 is commercially used in the industrial biotech sector by multinational companies, and the C1 technology is covered by 20 or more patents. The vast potential of Dyadic's C1 technology is to help speed up the development, lower production costs, and improve the performance of biologic vaccines and drugs at flexible commercial scales. The C1 technology is beginning to be recognized by the pharmaceutical industry. We are gaining momentum at introducing C1 to the pharmaceutical and biotech companies and governmental agencies, and have already attracted 2 top 10 pharmaceutical companies who are helping to fund initial proof of concept studies relating to how they may be able to utilize C1 to develop and produce biologic vaccines and drugs. We are positioning Dyadic's C1 gene expression platform as a potential healthcare game changer to drug developers and manufacturers, as well as governmental agencies in the U.S. and overseas. C1 can play a significant role in reinventing the development and production of biologics. If C1 is adopted by the pharmaceutical industry and governmental agencies, such as the U.S. FDA, as a potential alternative to CHO expression systems, we would expect that would lead to improved access and cost to patients and the healthcare system, but most importantly, save lives. Slide 4. The company has built an experienced leadership team, board, and advisors with 20-plus years of fungal gene expression experience and pharmaceutical industry experience. The company has engaged research partners and organizations who have the required experience and deep understanding of fungal gene expression and direct knowledge and hands-on experience working with, developing, and commercializing products using the C1 technology. Dyadic is laser-focused on further developing our patented and proprietary C1 technology into a safe and efficient gene expression system for human and animal pharma applications. In regard to our financial strength, we have more than sufficient capital to execute our research and business plans and believe we will not need to raise dilutive capital for many years, if ever. The company has a very strong balance sheet. As previously reported in our Q2 conference call, including the approximate $7.4 million we received from DuPont in July, we had $53 million of cash and investment-grade securities with no debt. The company recently initiated a second buyback program in August 2017 of $5 million. This is in addition to the $19 million share buyback program that was successfully completed in February of this year. Our stock is listed on the OTC Market Stock Exchange, OTCQX, under the symbol DYAI. On slide 5, the C1 platform has been commercially successful in the industrial biotech sector with companies such as Abengoa Bioenergy, BASF, Codexis and Shell, and DuPont, who on 12/31/2015 acquired our industrial biotech business and the C1 technology for $75 million. What these companies sought was access to the hyper-productive C1 gene expression platform that Dyadic developed. We've been able to demonstrate expression levels greater than 100 grams per liter with approximately 80% purity in a GRAS, generally recognized or regarded as safe, designation by the U.S. FDA for food and feed applications. Combined, these world-class biotech companies paid Dyadic in excess of $110 million in C1-related license deals, milestones, and equity for the C1 technology in our industrial biotech business. It's important on slide 6 to note that in connection with the DuPont transaction, Dyadic retained the exclusive rights to sublicense the C1 gene expression platform for uses in developing biologic vaccines and drugs for the animal and human pharmaceutical industry. We are entering the pharmaceutical market with a very powerful, industrially proven gene expression platform. If the science delivers on its promise, C1 will have the potential to dramatically lower biologic manufacturing costs, which we expect will create very attractive economic returns to the company and our shareholders in various forms of potential license deals, milestone payments, other collaborations, and/or the potential of joint ventures or the sale of the C1 technology for use in developing and manufacturing vaccines and/or antibodies and other therapeutic enzymes and proteins. Slide 7, C1 for biologics. On slide 8, we believe the biopharmaceutical market is an excellent opportunity in which to leverage our C1 technology. Biologic drugs, such as antibodies, make up the fastest growing segment of the biopharmaceutical industry and are some of the most expensive treatments. Therefore, they are placing an enormous burden on both patients and the healthcare systems globally. The 4 segments of the market that we are addressing with C1 are recombinant vaccines, which is projected to grow to $48 billion by 2021, new biologics, which is projected to grow to approximately $480 billion by 2024, biosimilars, biobetters, the non-glycosylated insulin alone projected to grow to at least $39 billion by 2020, biosimilars, biobetters, which are glycosylated proteins, which is projected to grow to $11 billion by 2021. Slide 9. We need to work with the pharmaceutical industry and governmental agencies such as the U.S. FDA to create and speed up the adoption of a new paradigm for developing and manufacturing biologics. A bottleneck exists in the development and manufacturing of biologics. The bottleneck is a result of low-yielding gene expression systems utilized today by the industry, underfunded development efforts for a more efficient next-generation gene expression system. The biopharmaceutical industry's reluctance in the past to utilize advances in science such as synthetic biology and genomics to develop next-generation gene expression systems for manufacturing biologics. We believe utilizing the rapid advances and ever-increasing affordability of synthetic biology and genomics, Dyadic is uniquely positioned to develop C1 into a next-generation protein expression and production system. Further, we believe the unique attributes of C1 will potentially create attractive research, licensing, collaboration, and other opportunities if C1 demonstrates the operational efficiencies, improved properties, lower costs, and reduced capital requirements for biologic vaccine and drug manufacturers. Slide 10. As a result, Dyadic is developing what the industry is starting to refer to as a CHO stopper. The industry has relied on CHO, Chinese hamster ovaries, for 30 years, and the need for a next-generation expression platform is significantly overdue. We and others within the pharmaceutical industry believe that CHO cells simply do not provide the output and productivity needed in the long run to dramatically lower the cost of biomanufacturing to produce affordable medicines for the global population. Dyadic's goal is to further develop C1 into a safe and efficient gene expression system to help speed up the development, lower production costs, and improve the performance of biologic vaccines and drugs at flexible commercial scales. Slide 11. As we did previously when commercializing the C1 technology for use in industrial biotechnology, we are reaching out to a broad spectrum of biopharma and biotech companies pursuing partnerships, R&D collaborations, licensing, and other commercial opportunities to leverage the value and benefits of the C1 technology for developing and manufacturing biopharmaceuticals. We have secured proof of concept programs with 2 top 10 pharma companies, and others are in the pipeline. Our reception from big pharma is accelerating, and consensus is long-term biopharma needs an alternative to CHO to meet the needs of developing, producing among other proteins, bispecific and multispecific antibodies, Fabs, non-glycosylated enzymes and proteins, and vaccines. Slide 12, C1 for biologics, the science behind the curtain. Slide 13. Let's talk now about the science behind the curtain. From a science perspective, the C1 gene expression platform attributes appear to be ideally suited to be applied in the development and manufacturing of biologics. The C1 cell line is a scientific anomaly compared to other filamentous fungal cells. The C1 cell line has unique morphology, which translates into better growth conditions and very high secreted protein yields, and has been used in industrial production for 20 years up to 500,000 liter scale. It's achieved 80% purity of the target protein secreted, which translates into greater retention of protein through downstream processing, lower cost synthetic media, no viruses like CHO cells, which eliminates 2 purification steps typical for CHO production, low pH viral inactivation, and 2 virus nanofiltration are eliminated. Wide pH and temperature operating conditions, which translates into more reliable and robust production processes. Shorter production cycle times, which translates into savings of nearly 10 to 14 days versus CHO for the process of seed flask to fermenter, and additional savings related to fermenter cycle time to 5 to 7 days, which are half to a third the fermentation production time of CHO cells. World-class protein expression up to 18 grams per liter of the target protein/enzyme have been achieved with C1. Slide 14. C1 enables shorter production cycles in comparison to CHO, which means a company can develop a new cell line in approximately 15 weeks, or a time reduction of greater than 50% versus CHO, and free up capacity. In a short period of time, C1 has already achieved initial non-optimized expression levels of 2 to 5 grams per liter and a IC to LC ratio of 1 to 1. Slide 15. As you can see, C1 has significant potential advantages as a production host for biologics. Now I would like to provide you an update on our research and development efforts with C1. Slide 16, C1 for vaccines. On slide 17, we have completed research that demonstrated C1's ability to express 5 different biologically active recombinant HAs, the flu vaccine. On slide 18, in an immunogenicity mice study, which was performed by Sanofi Pasteur with the C1 expressed HA New Caledonia protein, demonstrated the full-length recombinant HA produced in C1 from the HA New Caledonia strain showed excellent immunogenicity properties in mice without an adjuvant. On slide 19, additional data from the Sanofi Pasteur mice data study showed that the full-length recombinant HA produced in C1 did not induce any negative clinical signs in mice. No weight loss, no negative clinical signs during the experiment. C1 can potentially produce levels of up to 1 gram per liter of HAs and other antigens in 5 to 7 day fermentation, which means, performing the math, that C1 has the potential to supply the annual global HA strain needs against influenza with only 3 1,000-liter scale C1 fermentation runs. And that's 3 runs, 6 weeks of 1,000-liter, not 3,000-liter runs all year. That's amazing. Potential. Slide 20. We are also participating in a research program, ZAPI, in the EU, which is an R&D program sponsored with the goal of developing a platform suitable for the rapid development and production of vaccines and protocols to fast-track registration of developed products to combat epidemic zoonotic diseases that have the potential to affect the human population in this program. The original antigens were expressed and secreted in the medium in C1, and the preliminary results of a small mice test with one of the initial C1 expressed antigens within the program indicated that C1 produced antigen-generated an immune response in mice that protected the mice and did not have negative effects on the health of the mice. In addition, we have also initiated a C1 development program to express virus-like particles, VLPs, for antigen expressions, which is another potential emerging opportunity. Slide 21, monoclonal antibody expression. On slide 22, we are continuing to further develop and make improvements to C1 strains for the therapeutic protein development and production. As you can see, there are progressive steps in this progress to achieve our targeted protein productivity goal of 20 grams per liter and low proteolytic activity in half to third the time of CHO with much lower media cost without the need for viral inactivation and virus nanofiltration. The circled LC110 kern strain was the first step, developing a blank strain with a low proteolytic activity. The second step was loading the blank strain with a monoclonal antibody gene produced the LC110 MAB strain, which achieved mid-single-digit productivity with low proteolytic activity. The next steps are to further modify the LC110 C1 MAB strain to create the LCX, which is a future strain, at X times where we are today, that would deliver, we expect, up to 20 grams per liter of MAB protein productivity with very low proteolytic activity in half to a third of the time of a CHO cell with very inexpensive media. On slide 23, Dyadic and our research collaborators have experience with molecular capabilities, genetic tools, genome sequence and annotation, computational biology, and genetic manipulation, which are necessary to optimize the C1 strain for reaching high productivity and necessary functionality for targeted biologic vaccine and drug classes. We've begun the glycoengineering stage to further develop our C1 gene expression platform to form mammalian-like glycan structures. Further improvements on the horizon include further evaluating synthetic promoters and other genetic elements to drive higher productivity and to continue to glycoengineer C1 cells to express proteins with mammalian-like glycoforms in order to expand the areas in which we can apply C1 in the biopharmaceutical industry. Slide 24. I would like to provide an update on the status of our current C1 strains. They are demonstrating very promising initial results in their ability to express monoclonal antibodies. To date, we have demonstrated that C1 has expressed 100% of the tested MABs in our pharma research collaborations. MABs tested reached approximately 2 to 5 grams per liter in 5 to 7 day fermentation in one of those programs. MABs were integrated specifically to a hotspot in the genome, which means we can directly insert genes into a specific area, which is important for the process. The MABs were secreted to the media and properly folded, and the HC and LC ratio was 1 to 1. On slide 25, as discussed previously, the next steps for increasing the production level of C1 is by, among other things, identifying and developing better genetic elements such as regulators, transcription factors, promoters, secretion signals, etc. The synthetic promoters we are evaluating currently, we have tested synthetic promoters with 4 different core promoters using M-cherry as a reporter gene. The promoters were all functional and gave variable levels of expression. Amazingly, 2 of the synthetic promoters had higher expression than the C1 native promoter. As a result, production vectors with these synthetic promoters are under construction and being evaluated. On slide 26, this next slide illustrates what I said on the previous slide, that the expression of targeted MABs by synthetic promoters was increased by several folds by using synthetic promoters. Slide 27, screening of a C1 monoclonal antibody strain using random mutagenesis identified 25 higher producers out of 4,000 mutants, with one mutant reaching an 8 to 10-fold production increase. On slide 28, glycoengineering. Slide 29, as I said earlier, we had started already glycoengineering of C1, which will provide the formation of various glycan structures. Our goal is to have a library of C1 production host organisms with different glycoforms that can be used to dial up or dial down glycosylation to evaluate immunogenicity in vaccines and MABs. The results obtained so far appear to be in line with our expectations and glycoengineering development plans. On slide 30, C1 advantages and applications. On slide 31, we believe monoclonal antibodies developed in C1 will, among other potential benefits, save time and cost. After receiving a gene sequence from our collaborator, we project that the development time is now approximately only 15 weeks to create an initial non-optimized C1 monoclonal antibody strain with expected productivity of between 2 to 5 grams per liter in a 5 to 7 day fermentation. Slide 32. The next slide is a repeat of slide 14, but I want to reemphasize that C1 enables shorter production cycles in comparison to CHO by greater than 14 days or 15%, which frees up capacity, or 7 days or half to a third the time of the production time in fermenters. On slide 33, in order to shorten development and production cycles, the cost savings of manufacturing MABs in C1 could dramatically alter the economics. At any scale, C1 is expected to significantly lower OPEX and CAPEX costs. On slide 34, as you can see, C1 has vast commercial potential opportunities across a wide range of applications in the biopharmaceutical industry. On slide 35, in summary, as the aging population grows in developed and undeveloped countries, we and others within the pharmaceutical industry believe that the C1 technology has tremendous potential to help bring biologic drugs to market faster, in greater volumes, at lower cost, and with new properties to drug developers and manufacturers, and hopefully improve access and cost to patients and the healthcare system, but mostly importantly, save lives. If you'd like to learn more, please contact Matthew Jones, who leads our business development and licensing efforts at mjones@dyadic.com. Thank you. We'll now open up the calls for the Q&A. So I guess there's a question from Dennis Madden. How will the company build upon the recent DuPont project that just ended? How will this ultimately affect C1 development and technology? The answer to that is that the contract research organizations that we're working with have vast experience working with filamentous fungi. And in one case, the organization has vast experience not only working with filamentous fungi, but a decade's worth of experience of working specifically with C1. And those scientists are experienced in working with C1, not only on genetic engineering to overexpress proteins, but also fermentation optimization, and had worked on the project with Abengoa to commercialize over 100 grams per liter of protein in a 500 cubic meter fermenter. So they have the skills and the expertise and the talent to advance the technology, we believe, faster and further. The next question, are you working on any programs yourself or do you need partners? By Lee Alper. We're doing both. We prefer to work with partners, because, of course, they have certain needs that are unmet that we could potentially address to bring better, cheaper, lower cost biologics to the marketplace. And of course, funding research and development dollars is great. It helps us advance a platform that we could use not only for them, but for ourselves and other collaborations on the road. However, we are also, in addition to working on programs with others, as we mentioned, 2 top 10 pharma companies we're already doing research and development projects with, and we have others in the pipeline. We have our own molecules that we're working on, developing and moving along, that ultimately is proof of concept in terms of yield, performance, and quality to help us develop, license, and potentially sale of the technology, and potentially to sell those cell lines off in the future to partners. Next question we have is, C1 looks to be a tremendously powerful platform. Are you engaged with any pharma to perform POC? I think I just answered that question. The answer is yes. We are involved with 2 top 10 pharmaceutical companies to conduct research and development on their monoclonal antibodies and FCs. And we, of course, are talking to a variety of others. And we said we expect to land a 3rd top 20 biopharmaceutical company by the end of this year. Next question, I guess, is from John O'Neill. What is the latest update in the company's stock repurchase plan? I think I addressed that we have a purchase plan in place of up to $5 million, and we'll report on that in the next Q. If there are any additional questions, please submit them to the virtual booth, and we will answer those questions there. Thank you very much for joining today's conference call.